Good afternoon. Uh, uh, I already have sent you certain number of questions pertain to the lecture that I'm going to give this afternoon. Maybe you want to eyeball those questions only. Just go through those questions. I'm sorry it's not going to be a small group discussion. I'm not going to ask very many questions from you. And just before I'm coming or heading to Dominica, Dr. Ahmadi sent me a note that please give a talk on Oropouche virus. It is very timely because I show you the statistic that growing number of the people in South America and the Central America are coming down with this old virus. But we are learning about new behaviors and biological features of this virus. To be honest, when first I want to prepare myself for this talk, even I didn't know how I should pronounce Oropouche virus. I was thinking this is a French word, maybe. So, but I educated myself about this virus. I have not seen even one single case of this virus. We do have this virus among travelers, particularly who are coming from Cuba to United States. But myself, I have not seen even a single case of Oropouche virus. But I would promise you that this virus is in Dominica. So let me go through the timeline. So 1955, we discovered the virus. It doesn't mean that the virus is a new virus because 1955 is not a long time back. But we discovered the virus in 1955. Imagine when we discovered Ebola virus, filoviruses, in 1976, when we opened the genomic clock of the virus, we learned that the virus is more than 100 million year old virus, but just never had opportunity to breach to the human. The same story is true with Oropouche virus. Oropouche virus is old virus, but was an orphan virus. What I mean by an orphan virus was a virus among very marginalized community, very underserved, very remote communities, and nobody would care about those communities. Why suddenly, for instance, we learn so much about Ebola virus? Because Ebola virus from Liberia jumped to United States. Then suddenly a virus that we were not much interested on became very popular virus because a few of the American, a couple of Germans, a couple of really people from Europe, they did come down with this virus when they were traveling to Liberia or Guinea or Sierra Leone. This virus is an orphan virus. It was discovered in only one person, a febrile forest worker in a village in Trinidad, very close to the river named Oropouche. So we call it Oropouche virus. In a matter of fact, Ebola also, we call it Ebola because we learn about Ebola virus among a community very close to a river named Ebola. So it is not unusual to name the viruses after the place that they have been discovered for the first time. And this is the basically the map. You would see there are plenty rain, plenty forest, and you see the basically Oropouche River. And this is the reason that we are calling this virus Oropouche virus. This is more landscaping. And when over the Saturday, I was basically, I went to different parts of this island. In a matter of fact, I saw a lot of similarity between the ecosystem of the Oropouche River and the landscape of Dominica. So is the best place basically for this virus also to reach to the human. Unfortunately, it's not only the first one of the arboviruses. When we call arbo means arthropod, means insect-borne viruses. This means that the insects, they carry this virus. Arbovirus infection in Latin America has a long story. You are familiar, for instance, with dengue fever, and then you're familiar basically with chikungunya, with Zika, and now Oropouche virus is a new addition to the Latin America arboviral infection, uh, and is a major challenge in diagnosis because all of these viruses, when they come to a clinical presentation, they are almost overlapping each other. There is no any specific feature of the dengue that makes dengue distinguished from Oropouche. But I will go over the clinical manifestation of this virus also in depth. So prior to 2000, we did know about the virus and it would give some outbreaks here and there, but not major outbreaks. But then you can see gradually this virus turned corner and behaved like a new virus. Many often I may, during this talk, if you ask me a question, I may say, I'm sorry, I don't know. Because we are learning about this virus. It's just we are touching the mystery of this virus, what is very mysterious virus. Biological behavior of this new outbreaks that we are dealing with in Latin America is so different from the nature of this virus in 2000 or before 2000. And this is the new outbreak. So I showed you some background that this virus is very old one, but look at these new outbreaks. The new outbreaks, we are learning about them from late 2023. And now from June of 2024, there are many people in Cuba that are coming down with this virus. Cuba never reported any case of the Oropouche virus before. And now we have so many of the outbreak of this virus in many of the communities in South America. So the new outbreak focus on 2003, 2004, 2005. As of August of 2024, almost 8,000 cases of this virus has been reported in Bolivia, Brazil, Colombia, and Peru. And for the first time in Cuba. A few cases have been reported in Europe, and also probably 100 of them have been reported in the United States, but has been among travelers. And majority of these travelers, they are coming from Cuba. Overwhelming majority of the patients in the United States, in a matter of fact, are among travelers coming from Cuba. But there is no infection acquired by this virus inside the United States. And this is the map, and there are obviously color code. The dark color means the outbreak now in these countries. So you see mainly is Brazil But then you can see Caribbean islands. And then the lighter color, which is on the southern cone of the South America, this is where the old outbreak of this virus has been occurred. So the virus, in a matter of fact, if you look very carefully, you can see the virus is moving from the southern part of the South America toward the northern part of the South America. And just to show you a couple of points, in Argentina, the virus is very well known since 2005. But if you look in Haiti, you can see it is since 2014, for instance. And look at Barbados, which is very close to you people. It has been described in 2024. And then if you look at the number of the countries like Cuba, like Colombia, you will see 2024. So for the first time, we are learning about this virus in part of the America that this virus never has been reported. But we never had very good diagnostic tests. We never really look into this virus very vigorously. So it might be there a sort of the outbreaks and even the clinical manifestation of this virus that I'm going to show you now. It maybe happened even 30 years ago or 50 years ago, but nobody has diagnosed Oropouche virus those days. Because as I said, Orovirus, Oropouche virus is an orphan virus. And this is distribution of the patients with this entity, this virus in United States. And you will see majority of them are in Florida. And these are all among the travelers. We have not proven any single case of Oropouche virus acquired in North America. We have dengue in America. We have among travelers, and also we have among the people who never travel. We have chikungunya in United States. We have Zika in United States, but we have not yet identified any cases of the Oropouche virus. There are almost 101 cases, and unfortunately, this slide is maybe about 2 weeks old, but probably by now we have a little more number. But overwhelming, as I said again, all of them are among the travelers. And this is interesting slide and also very misleading slide. This means that overwhelming majority of the Oropouche virus are among the travelers that they traveled outside the United States to the endemic places during summer. But there are so many other explanations for this curve. Could it be the number of the travelers that they travel from the United States outside the United States are more during the summer? Yes. If you look at this, for instance, in November, we never had any single case of the virus among any traveler in the United States. But the major question is how many people are going outside the United States to endemic areas in November? I am here in January. I'm not really a common traveler. The majority of the traveler, they come to Caribbean islands during the holidays. They come during the summertime. They're coming during really the Christmas time. But this means that among the travelers, that overwhelming majority of them, they travel during the summertime. Obviously, they bring the virus home. And this is the slide of the cartoon of the virus. So is ball shaped, is cylindrical. And then look around, you would see there are so many of the cell membranes, epitopes. And we have 4 0 bar of this virus. Dengue also has 4 0s. But dengue, if you come down with the first attack of dengue with type 1 dengue, now you come with type 2 dengue, then is when you are coming down with severe form of the dengue. Dengue, the first attack is okay. This is the second attack of the dengue that is giving you hemorrhagic fever, is going to give you redistribution shock, for instance. In dengue, if you acquire type 1 of dengue, it doesn't give you any protection against type 2 dengue. So the vaccine that we made for dengue has component from 4 serovars of dengue. Dengue type 1, dengue type 2, 3, and 4. We combine them and we make a vaccine for dengue. But Oropouche virus, though has 4 serovars, but if somebody comes down with one of the serovars, that would give that person immunity for the rest of their life, his or her life, and there is definitely cross immunity against these serovars. The other thing that I want to show you in this slide, and it is very important, look at the segments of the nucleus. There are 3 segments. There are circular and there are 3 segments. If you look at the influenza, it has 9 segments and they can mix. And that's the reason every single year you're dealing with the newest strain of the virus. This is the reason that if you vaccinate me against the influenza this year, it is absolutely useless for the next season, because the next season I'm dealing with a new serovar of the influenza. If the vaccine that I have, it is not going to protect me against the bird flu, because the epitopic features of the bird flu is completely different from the influenza virus that currently circulating in different parts of the world. And this has 3 segments. And when I come to biological features, the new biological features of this virus, you need to remember entire information of the genomic information of the virus is not on one piece of the gene. They are on 3 segments. And one cell is going to be co-infected by 2. Then imagine you have 6 pieces and you can combine these 6 pieces into different form of the virus. So Oropouche virus has capacity of changing biological genomic identity of each other after some time. And also, we do not know yet how the virus interact with the cell membrane of the cell. If I'm coming down with the virus is going to get to my body. How the virus is going to get into the cell, we don't know yet. We know that LRP1, which is obviously one of the lipoprotein on the cell of the human cell, is very important molecule for the virus to land on. But we don't know exactly how it happens and how it gets into the cell. What we know is going to go through the cytosol. So this is the cytosol. So if you look at the virus, this is the virus is coming to the cell membrane, and then you have this vesicle, cytosol, or endosome, and it's going to get into this one. And then obviously the nucleus or the genetic information needs to be released. It's going to be released. This is genetic information of the virus. And now it's going to go through transcription and replication. And then is going to make all of the compartments of the virus. And then all pieces of the different part of the virus is going to again go to another endosome, and assembly is going to happen. And then the virus is going to get out of the infected cell and is ready to go and infect another cell. So this is the electron microscopy of the virus, and this is the 3 segments, a small, medium, and a large. And you need to remember that this virus is negative sense RNA virus. So when I say negative sense, or when I use positive sense, there is no positivity, there is no negativity. This is nomenclature, which is very confusing. But when we say negative sense RNA, means the RNA of the virus can't be the template for multiplication. it has to convert to a template that becomes the template of replication. So some of the viruses, when they get into the cell, they are ready to multiply. This virus getting into the cell needs first to become positive sense, and then the positive sense can become the template of the replication of the virus. And this is the major difference between this virus and the other arthroviruses, which are common in this part of the world. You can see dengue virus here, you can see chikungunya virus, and you can see also Zika virus. All of them, they have one single strand or one basically segment genome. They don't have three segments. This is the reason dengue virus, for over 50 years, 100 years that we know about this dengue virus, the biological behavior of the virus has been absolutely the same. And clinical manifestation of the dengue virus 50 years ago was absolutely clinical manifestation of dengue in 2025. But this virus has potential. I say potential because we have not yet proved how this virus changed and how this virus clinical manifestation or even causing a big outbreak, it has been changed. In how? What is the molecular biology behind that? We have not yet explored that, but this is just an assumption based on obviously scientific merit of the virus, three segmented virus, and the chance of the co-infection and the mixture of different basically segments coming to a new form of the virus. I put a lot of words in these slides very intentionally because I knew that I sent and you all are going to have access to these slides. Because I put a lot of words for medical students when they want to basically review this lecture, they shouldn't have any problem because they can read basically slide by slide and it is completely comprehensive. But I want just to go over some history of the mosquito adaptation. Imagine when we were hunter and gatherer. So make you yourself in 40,000 years back or 25,000 years back. Obviously, the mosquitoes, they didn't have enough number of the human to maintain themselves on the moon. The insects, they were living on canopy of the trees. And on canopy of the trees were enough water, and it is enough water now, and is enough of the mammals, monkeys, imagine non-human primates, rodents, insects to circulate themselves among them. Mosquitoes stay on the top of the canopies, and the virus also stay on the top of the canopy. Mosquito would get the virus from one monkey and then would give it to another monkey on top of the canopy. And the humans were few and they were living gradually left the forest and they came basically to the farms. And then the mosquito became smart. Say, okay, now I have enough number of the people. Why I shouldn't take advantage of the people? Why not I should adopt myself to human? But mosquito needs now to come down from the canopy and leave the forest behind because the human gradually, for instance, 5,000 years back, we had civilization, two big civilization in the South America. And we had major civilizations in the other parts of the world. We had cities, we had towns. So now the mosquito needs to learn how to breathe because mosquito needs water. And this group of the new mosquitoes that adopted to the humans in the cities, they take advantage of tiny amount of the water. Do you have any flower at home? Do you have any flower or plant pot that lead under the pot as a little water and has 3 days of the water or 4 days of the water? The mosquito needs that much of the water to put lay egg and then the egg matures and then become another mosquito. Or tires, spare tires. We throw them all over the town. Rainwater is going to get into the tires and it's going to stay in the tires. And the mosquito is going to basically be on the tires. Now, if I'm coming to the Oropouche virus, you see you have the sylvatic cycle and then you have the urban cycle. This is the urban cycle that makes the virus maintain, replicate, substantiate. And a virus that is going to go through changes with three segments, God forbid, you do not like that virus to multiply. So if that virus is going to find chance for multiplication, diversity is going to be the outcome of that multiplication. Each son or daughter is going to have new traits, completely different from the parents, is going to completely behave differently. So now, if you look at this one, then the other thing that you look, you will see the reservoirs. There are not only one or two or three. Reservoirs means the virus that lives in body of another host. Human is a reservoir, could be the reservoir. But look here, we have monkey. We have basically three toes sloth, for instance, and we have the other primate. And then we have question of even birds and rodents. Do you think we can eradicate this virus? No. Did we eradicate the smallpox? Yes. Why we eradicate the smallpox? Because there was no any reservoir except human. The virus could go from human to human. So we find very nice vaccine. We put ring vaccination and we interrupted the cycle of the virus and the smallpox now is history. Two countries, they have some of this virus in tubes, present, one in Russia and one in the United States. But still, we do have the smallpox virus, but in two labs, one in Russia and one in the United States. But it is not natural way. Can we eradicate, for instance, polio? Yes, we can eradicate polio because there is no any natural reservoir for the polio. Already, I can say that we eradicate polio in majority of the world, but not in Pakistan, for instance. And the one that we have sometimes report of polio basically in somebody in United States, these are vaccine derived basically polios. These are not the wide virus polios because polio, we have two different vaccines. We have live vaccine and we have, as you know, the kill vaccine. The live vaccine sometimes is going to retrograde and become viral. And now this is obviously, there is no way that we can eradicate this virus. Now somebody goes to the forest and then is going to be infected, is going to be bitten by a mosquito, for instance, that is infected with the virus. This mosquito already has fed, for instance, on a monkey's blood, that the virus was in the monkey blood. So now it's in the mosquito, and then mosquito jumps in the human. And this human lives in the town, now brings the virus to the town. Now this is the man, the human, which has the virus in the blood. But here's something that you need to remember. The major culprit of the circulation of the virus among human in towns is not mosquito, is midges. And I saw so many of them on Saturday evening. So in making the story short, so obviously the virus is in wild, is going to get to the mosquito, and the mosquito is going to give it to the human, and human is going to bring it to the town, and then is going to get basically to the midgets, and then the midgets are going to spread it among the humans. So this one is very important lesson to remember that the cycle of this virus is so different from the cycle of virus of the, for instance, malaria. Because malaria, you have the Anopheles mosquito, and that's it. But here you do have two different types of vectors. So when I say vector, so means basically something, animal, a creature. It's good now. It's okay. And then obviously when it gets to the human, then obviously, another insect is going to carry it out, but not malaria, for instance. So this is a short summary of the life cycle of the virus. And this is show you the reservoirs. There are multiple reservoirs. There are not only one or two. And this is the vector. And then you can see mosquito and midget. And if I show you the midget, and then this is going to be mosquito, then you can see the size. So mosquito net is phenomenal for the mosquito, but midget, they can easily pass the mosquito nets. So the mosquito nets are not going to help you very much if you want to prevent the outbreak of the Oropouche virus. You can see the size of the midget, just you will see tiny, very small. I'll show you, this is the mosquito, and this is, you can see the different in size, and show you a couple of slides, but you can study them later on, that obviously midges is not only carrying the Oropouche virus, they are also carrying some worms. They are also carrying so many species of the bird malaria, for instance. And this is another example, but you can read for yourself later on. But obviously, tiger mosquito, this is the one which has adapted itself to the urban condition, easily can move with the human from one community to another community and can multiply so easily in a small amount of the water which stands by your house. And you will see dengue fever, yellow fever, Zika virus, and chikungunya virus. All of them can be carried by the by tiger mosquito. And now we are learning that maybe this is vertical transmissible disease as well. It's like Zika. We do not have yet much of the literature on that, but I'm going to show you some literature to show you that is very concerned that if the mother is going to be infected by the virus may pass through placenta and then may get to the fetus, and then it's causing a lot of congenital anomaly. And it causes abortion, miscarriage, basically, deformity of the central nervous system. And this is one study, but obviously is not proving that this virus is vertically transmissible. When I say vertical transmission, I mean from mother to the child to the placenta. And this is the study which was published in Lancet, Infectious Disease, and they checked for the orovirus IgM among the newborns, 68 newborns with microcephaly, and they did identify in 6 of them the antibody against the Oropouche virus. And this study is obviously done in the place that Oropouche virus caused the big outbreak. And then one of these patients, which is very concerning, this infant died and had orovirus RNA and antigen in several tissues, including the brain. So this is a concern. This is definitely now the people need to explore this possibility and to be certain that this is vertical transmissible or is not vertical transmissible. But the CDC is considering this one vertical transmissible till otherwise. I will show you some of the recommendation from the CDC. For instance, the pregnant woman should avoid to go to any country that has the Oropouche virus. But unfortunately, the data that we have is very limited at this point of time, and we cannot really give a general statement or make the final point that yes, this is the vertically transmissible and virus. But the healthcare provider should be aware of the risk of vertical transmission, and they have really to educate the patients and the travelers very, very carefully. And this is now completely new. And I will show you some literature that for the first time, we identify the virus in the semen of the person who has been infected with this virus. We knew that, you know, some of the viruses, they easily get to the, uh, you know, genital system of the male and the female. For instance, Ebola virus is the classic one. Ebola virus, there are literature in New England Journal of Medicine. Somebody got the Ebola virus, went to Germany, and then passed it to his wife. And then they identified the PCR of the virus in the semen for almost 5 years. And also, as you know, when the epidemic of the Ebola virus in the west part of the Africa was confined, after a few months, there was a minor outbreak of the Ebola. And the source of that minor outbreak was a woman, a nurse. So the women, they can pass the Ebola virus and they can transmit it to the man. But for this virus, we do have limited, as I said, limited info, but we have not really yet proved whether this is sexually transmittable or sexually not transmissible. Because finding the virus in the semen is something, and being sexually transmittable is completely different story. Don't think if the virus shows itself in the semen, can all the time be transferred to the partner. No, because between the virus and the mucosa of the partner, there are so many other biological, immunological contributes to the defense and to the basically uh, allowing the virus to invade the body. And one, this article is very worrisome because they showed that dengue and Oropouche virus, they were co-infected in one person. But more importantly, when they look for the virus in blood, they saw the virus is in blood of the infected person for almost 36 days. And the person was completely asymptomatic because this virus would give you acute illness for a short period of time and you recover or you come down with severe form of the virus. But the person recovered, but they could identify the virus in blood of this man for 36 6 days after the onset of the uh, of the symptomatology. So this person is saved in Cuba. You can see July 1929, and then you can see basically symptom onset, and then from symptom onset, then you will see what they checked, and they look into the whole blood, they look into the serum, urine, semen, and then obviously check in different intervals, and then you can see easily if you look at the whole blood, you would see that it is present for a long period of time. But it was not only just the PCR showed the virus in blood, and this shows you the cycle threshold. I'm not going to basically take your time to explain what cycle threshold means the concentration of the virus basically in blood was substantial. But more so when they got this virus from blood and they put it in cell line, it was the cytopathic. On the other hand, the virus was pathological. It was not only just a piece of the virus, because when you have, for instance, you suspect COVID in someone and then you send a nasal swab, you're checking for the PCR. You're checking only for a piece of the virus. It doesn't mean that the virus is in the nose. It means a segment of the virus is in the nose. It doesn't mean that the person is contagious, but means that the person has the specific epitopes of the virus in the nose. But here, the PCR was positive, but also when they culture it, they grow the virus, and also the virus was cytopathic. So it's very worrisome. But for you need to remember that this virus is not a spread from person to person through coughing, sneezing, or touching. It only needs mosquitoes or midgets, and they have to come to the patient who is viremic, suck that blood, and then go and spread to someone else. So if I'm going to Cuba, for instance, and coming to the United States, I'm supposed to keep myself away from the insects for 6 weeks. Because if I do have the virus, I do not like to give it to the local mosquitoes and midgets in United States. If somebody has proven Oropouche virus, it's going to be also advised to do not have sex, or having sex has to use condom for 6 weeks. Now coming to the clinical features. This is one of the viruses that overwhelming majority of the people with this virus, they have symptoms. This is completely different from other viruses that maybe like SARS-CoV-2, practically speaking, 40%, 50% of the people who are infected, they are asymptomatic. But overwhelming majority of the people with this virus, they do have symptoms. Incubation period is very short, is about 3 to 10 days. And obviously it starts with abrupt fever, but nothing really specific to this virus. Look like influenza, it look like dengue, it look like, another virus, myalgia, chills, arthralgia, and then photophobia, dizziness, eye pain. All of these things are really not specific. And the duration of the disease is very short, is 2 to 7. But another thing that we are learning about Oropouche virus, that unfortunately relapse is very common. So the person is going to do completely fine, and then after a few days, 2 weeks, the person is going to come down with the same manifestation of the original infection. That's the reason that paper that showed that the virus is going to be persistent in blood is very meaningful. This means that the virus has found a reservoir inside the body, like HIV, all the time in prostate, in ovaries, in the central nervous system. These are privileged sites of the virus. Immunity cannot get rid of the HIV in the central nervous system, cannot get rid of the HIV in the prostate, or in the ovaries, or even in so many other places in the body. So human become reservoir of the virus. And 60% of the patients, a few days or even weeks later, they did come down with the same symptomatology as before. This is the rash of one of the patients, looks like rubella, and usually it starts from throat and then goes to the extremities. So it's central, and then from center it spreads to the periphery. And now we are learning that unfortunately is neuroinvasive disease, is neuroinvasive virus. Do you know a couple of neuroinvasive viruses? You would say the classic one, probably rabies. 100% of the time fatal, 100% of the time neuroinvasive. What about polio? Because you know polio very much. The polio, we have three types of the polio, type 1, type 2, type 3, but it doesn't matter. 1 out of 1,000 to 1 out of 2,000 of them, they come down with paralysis. The rest, they do fine. But still, we call poliovirus neuroinvasive virus. Though only 1 out of 2,000 of the people who come down with poliovirus, they come down with polio. That's the reason the people who are against vaccination, they are all the time making this point. 2,000 people, they come down with the polio, they are doing completely fine. But then we say, well, listen, get back to the history. See, in 1954, what happened during the epidemic of the polio? How many of the kids, they acquired the polio in swimming pool? And we have to close swimming pools in the United States. And even our president that time was a victim of the polio. But in this one, 4% of the patient will develop neurological symptoms. And this means meningoencephalitis or meningitis and fatality from brain edema and brain inflammation. And unfortunately, during this stage, you cannot recover the virus in the CSF. You are most depend on the serological marker for the proof of the neuroinvasiveness of the virus. And the Guillain-Barré shouldn't be new to you because you know the Zika, particularly during the Zika season. You know, I was in Dominican Republic and I was walking through one of the intensive care units and they had 12 beds. 10 of the 12 beds, they were the people who were completely paralyzed by the Guillain-Barré of the post-Zika. And post-Zika, never forget that the virus here is not neuroinvasive feature of the virus. This is the molecular mimicry. The body makes antibody against a specific part of the epitopes of the Oropouche virus, and that antibody is going to cross-react with some component of the Schwann cells. That's the reason for Guillain-Barré. Is similar to multiple sclerosis, for instance, is more of the mimicry, is not the virus invading the neurocell to give you the Guillain-Barré. But there are now multiple literature on Guillain-Barré as one of the manifestation of this virus. Fortunately, so far, all of the people who come down with Guillain-Barré, they did fine after a few weeks. They survived, and there was no any neurological remnants of the Guillain-Barré in this group. So prognosis is good, but you should have ICU, you should have ventilator because they may need 2 weeks of, for instance, completely being on ventilator before finally they fully recover. Unfortunately, diagnosis, I'm going to talk a little more because some of the students may need the scientific info on that. But in places that this virus now is blooming, basically, we do not have any diagnostic tests. CDC does. But CDC is in Atlanta, United States. I do not think any lab in South America, maybe Brazil, probably they do have the lab. I think really, I learned that in Brazil they do have lab that can make the diagnosis. But you people have to make the diagnosis based on clinical picture, which is nothing really specific. Looks like another dengue, looks like another Zika. So, but history of the travel in United States would be very helpful, and activities leading to risk of possible exposure is also very relevant. I want just to make myself as an example for you to understand the complexity. Saturday, I went to see one of the hot springs, and it was, I think, almost 6 o'clock that we got to that place. And we went down, and then there was some light, and me sat basically under one of these lights, and midgets just were everywhere. And I came probably with more than 20, 30 bites. Now, the question is, what is the chance that I'm going to come down with Oropouche virus? First, you will say midget, midget. But yes, I acquired bites not in town. I acquired bites in forests. Mosquito bite was very dangerous in that place, not midget bites. The second, how many percent of the midgets in the town they are infected? This is something good for medical students. We don't know in this part of the world how many percent of the midgets they are carrying the virus. There are 2% of them, one out of 1,000 of them, or one out of 100,000 of them. If medical students looking for a research project, serological studies and surveys are the simple form but valuable form of the research. Do we know how many percent of the mosquitoes, how many percent of the Egypticus is carrying the dengue or Zika virus in this place? How many people in this community of 70,000 people, they already have serological marker for the Oropouche virus, or for dengue, or for Zika? These are very valuable information that at this point of time we do not have. The lab values, they are not very specific, a little of lymphopenia, the elevation of this, CRP. These are very, very non-specific and of no use. But you need to remember that thrombocytopenia may happen, and hemorrhagic fever is one of the manifestation of the Oropouche virus. And message is very clear, or the lesson that you need to remember is very clear. When you have a patient with any of this group of the viruses, please never use non-steroidal anti-inflammatory medication. No Motrin to this group of the people. No aspirin to this group of the people. Because you are going to increase the chance of bleeding in this group. So obviously we can do blood culture, we can do basically urine culture, we can do the RNA uh, in saliva, in urine, in blood, in the serum. We can check the IgM, which comes first, serological marker, or we can check the IgG. But I think really these don't have no value really in places that this virus is very common. And I told you for neuroinvasive disease, though we are all the time checked for the PCR in the CSF, but many often we do not have really the virus in the CSF. And CDC has all of the available tests, and we can request or we can send the sample to the CDC and Atlanta, and they can definitely run the test for us. Management, avoiding non-steroidal anti-inflammatory drugs, avoiding unnecessary treatment and procedure, and monitoring the patient for severe symptoms or complications. And obviously, we need to educate our patient about the vertical transmissibility, about transmissibility of the virus through the bites, and also for blood transfusion, for instance, they should not really give any blood for 6 weeks at least. If they want to donate semen, for instance, they have to really also uh, wait for 6 weeks. There is no any specific treatment, is all supportive care. And this is a misspelling here, rest, fluids, and use of non-steroidal anti-inflammatory, basically medication analgesics. Use Tylenol, acetaminophen, for instance, for the fever or the pain. Okay. And also, this is only one notion that now we do have. Many of the, not many, several of the technicians who have been working with the virus on the blood sample of the virus, they got needle stick and they came down with the virus. There is no vaccine as we were speaking, and the best way to protect yourself is bug bites. The repellent that you use, which is great for mosquitoes, they are not very great for midges. That's another problem. So the midget is the key insect in transmissibility of the virus. Neither mosquito nets and nor the repellent that we use, they are very much effective against this group of the insects. And this is obviously details on what we need to do if somebody has the virus, but prevent a spread through sex. If somebody does have the orovirus infection, needs to stay away, having no sex or having the sex with condom for the time. And then obviously, if somebody comes to United States or to another place from endemic place and has sign or the symptoms of the virus and has proven or virus obviously needs to be very careful to be not beaten by mosquitoes or midgets for 6 weeks when is in United States. The final part of my talk, which is very brief, but I'm going to basically talk a little about the new manifestation of this virus. On the other hand, I'm going to compare and contrast the old outbreaks of the Oropouche virus versus the new outbreak. Obviously, the explanation for this new behavior maybe is the change in the human behavior. Maybe we are knocking down the forest too often. Maybe we are intruding into the ecosystem of this virus. So ecological changes are definitely the key. And also never forget the new viral biological changes because of the three segments and the chance that is going to mix. I'm only showing you a couple of really basically slides to tell you the differences. So you would see the previous outbreaks, they were very much limited in circulation. They were in very specific part of the world, and they were more basically focused on sylvatic cycle. But now the new outbreak is in big cities. It is not anymore in the rural places. The old disease was benign. We have no report of death, for instance, from the old outbreak. It was mild, with headache, with muscle pain, nausea, and occasionally we had meningoencephalitis. We did not have any report of the vertical transmissibility of the virus. We didn't have any report on teratogenicity of the virus. But now this is one of the new articles, and you can see it's fatal Oropouche virus infection in non-endemic region in Brazil. And the two people who died from this virus, both of them they were completely healthy and they didn't have any comorbidity or underlying immunodeficiency. They were healthy, had robust physical fitness, but they did come down with this virus, and both of them they died. So the new virus then has a few new biological property that you need to remember. Clinically, it is more severe. It may kill. The third one has been reported in children for the first time. The fourth one is vertical transmissibility of the virus that under scrutiny at this point of time. And the final point that you need to remember, it is teratogenic, or we believe it is teratogenic. The impact is a lot, already overwhelmed Latin Americans, already a stretched healthcare system. This is going to be very demanding for eradication or control of this outbreak. And there are many unanswered questions that we need to answer gradually. If the people infected with Oropouche virus can spread the virus through the sex, though we have the virus in the semen, but can it be transmissible sexually? If it can be spread through the sex, how long people might be able to spread Oropouche virus through sex? If a spread of Oropouche virus during sex could cause a risk to pregnancies, if the virus found in vaginal fluids, what would be the impact on that, on sexually transmissibility of the virus, on fertility of the man, for instance? Obviously, CDC has plenty number of the recommendation for all of these potential problems. Future direction, obviously, we need a simple test be available in most of the countries that your resource limited. Unfortunately, we do not have any treatment for this one. Hopefully, we need really a medication for this disease. And obviously, we need to put very nice surveillance system in place to pick up the virus very quick. Because if you want to confine the outbreak, obviously you need to know where is the virus and how it behaves and what is the scope of the outbreak. And the key points that you need to remember, this is my final slide, is Oropouche virus is transmitted primarily by biting midges and some mosquitoes. Mosquitoes roll in forest and obviously midges in towns. No cases of sexual transmission have been reported, but the virus has been detected in semen. Oropouche virus has been reported in parts of South America, Central America, and Caribbean. In June 2024, Cuba reported its first confirmed virus. Oropouche virus disease typically present as an abrupt onset of fever, severe headache, chills, myalgia, and arthralgia. There are no vaccines to prevent, and there is no medication to treat this virus. I stop here, and I would be delighted to take some questions from audience. Yes, please. I'm sorry. I what is it? Oh no, that's my ears too. Really, I'm congested, and I'm also 76. So combination of the hearing deficiency and
Yeah,
all.
I was just asking about the NSAIDs. Were you saying that to avoid non-NSAIDs for treatment or for?
Absolutely.
Yeah.
Because non steroidal anti-inflammatory medications, they interfere with the platelet function. So if the person is already susceptible to bleeding or has bleeding tendency, then obviously you have to avoid any medication that is going to interfere with the platelet function, which are so important in blood clotting. Yes.
Please, I'd like to know how fatal it is, like the mortality rate, and also why is it that up to now there's no vaccine? If it has different variants, can it kind of be compared to the influenza so that we can have every year new vaccine? So, are they still doing some research about it?
Thank you very much for very important question. So, um, so why, let me tackle that, why we do not have yet a vaccine? Because we know that this virus can be fatal. How fatal this virus is, we don't know yet. We have case reports. We have two people who are very healthy and they die. We know it is neuroinvasive, and also neuroinvasiveness can be fatal. But to know the fatality rate, we need to follow the pandemic or epidemic and outbreaks, and then do very good surveillance tests, identify the people, follow them, and then see what will be the immediate consequences or the late consequences. And we are just learning about these outbreaks. As I showed you, the new literature just comes at the end of 2023 and then during the 2024. For vaccine, you need to remember there are a few challenges for vaccine. One, maybe biological challenges like HIV. We know about HIV for 50 years. We have tried multiple really way to make vaccine against HIV, but biological behavior of the HIV will not let us to overcome the challenges. United States alone probably has spent billions of the dollars to explore, to identify a vaccine for the HIV. The second thing which is very important is how relevant that virus is to wealthy communities. Ebola virus or Marburg virus, which is the cousin of the Ebola virus, was identified in Marburg town in Germany in 67. We even knew that it's easy to make the vaccine. But the question is how we can sell that idea to a drug company, to a company to make the vaccine. that is not going to bring any profit. Because this vaccine, if somebody makes a good vaccine for Ebola, that they did, Russian made phenomenally vaccine for the Ebola virus, as well as the Americans. So we have two very powerful vaccines for Ebola virus. But it was made after 4 decades when the epidemic happened in the western part of Africa, and 30,000 of the people they came down with the virus. Previously, the outbreak were 20, 50. The major outbreak in Gulu in Uganda was 495 people. So, but then American came down with the virus, German came down with the virus, then obviously attention suddenly changed because now is a virus that is threatening the life of the wealthy people, non-marginalized people. Marginalized people, who cares about them, unfortunately. That's the reason I said the other day in the White Coat ceremony to the medical students, if you are here for underserved community, you're welcome. You're welcome. You're genius. You have a lot of energy, good questions. You pick up the question very right. You need to become the director of the National Institute of Health. You need to become one big key in United Nations, one big key in WHO, and change the direction of what we see, unfortunately, at this point of time. Zika virus, for the first time now, we have a vaccine, very good vaccine for Zika virus. Probably tomorrow afternoon, I'm going to talk about Zika because I think my time is over now. But I do prepare for you another talk about Zika, but mainly focusing on basically the new powerful vaccine that is very, very helpful. And the people who are living in this community for more than 6 months, they all basically are eligible for receiving the new Zika vaccine. But when we learn about Zika virus was 10 years ago, and the Zika virus was not only the virus of the marginalized group, because we did have so many of the patients in Texas, we have so many of them in Florida, we had so many of them in other parts of the United States. It was in the semen of the people. We learn about sexually transmittable disease. We proved that this is the vertically transmissible virus. It became our problem. Then we tackled them. As long as any illness is not the problem of rich, rich is not going to pay attention.
Yeah. Okay, sorry. The male patient with the with the disease being detected in his sperm, was it a child? Was it a grown up? And how did they come about determining that it was in his semen versus somewhere
Oh,
else?
how they determine the virus in the semen?
Yeah.
Oh, okay. Obviously, what we do usually first we go by PCR, and the PCR we have a primer, and the primer is going to be very specific for some specific epitopes of that virus. So they identify the virus, at least the pieces of the virus in semen. Then we culture. The culture, I'll give you some example because the culture sometimes is hard, sometimes is easy, but the barriers are going to be very difficult. For instance, if you want to do a study on Ebola virus is easily you can culture it. But Ebola virus, if you get to you, if I'm scientist, I'm playing with the virus, I'm trying to culture the virus, and if I basically the virus breach to me is 100% fatal. So I need to do it in a lab that we call it level 4. So that is very sophisticated, and there are only few level 4 lab even in United States. We have a couple of them in Atlanta. Basically, Army has one, and a state of Connecticut. I do not think anyone has level 4 lab. SARS-CoV-2, the virus easily you can culture. So varicella, is very susceptible cell to the SARS-CoV virus. But SARS-CoV is very invasive. That's the reason you need level 3. Even before we learn about the SARS, because we were remembering SARS-1 and SARS-2, because they were very fatal, obviously we were using even level 4. For this virus, we can use really level 2 of the, so we can easily then send the semen for the culture, and then we find the virus is cytopathic. Then we know that the virus is live virus and pathogenic virus in the semen. But whether it's going to be sexually transmissible or not, there is, you have to wait. And that's the reason, for for instance, for the Ebola, I told you that somebody acquired the virus, a man, and went to Germany and then passed it to the wife. And then we got the virus and then we sequenced the virus and we see this is the virus that the woman has, is the same virus that the man has. Because sequencing is fingerprinting of the virus and we can easily say who gave this virus to whom. Basically, we can track the epidemiological track of the virus by sequencing of the virus. So, thank you. Yes. And I'm sorry that I missed your questions. Oh, oh, this is my guess because you people need to study that. But I believe the midges are more important because they transmit the virus among us in the big cities. Mosquitoes, they transfer the virus in a small, tiny communities in forests. But assume right now I have the virus in my blood and I'm not going to keep my skin away from midges. I'm going to infect the midges. And the midges, they need blood. They need blood. All of the mosquitoes, midges, all of them, when they are pregnant, when they have eggs to mature needs iron. So this is the female mosquito that bites you. The male mosquito has nothing to do. Male mosquito is much of a happy with flowers, fruits is going to be the source of the basically material nutrient for the male mosquito. But the female mosquitoes, female midges, when they are pregnant, when they have eggs, the eggs needs to mature and they need iron. That is when you're going to get blood. And they eat blood not for themselves. So all the time I show sympathy toward these female insects because they are protecting their own children. So the midget, I think, has more of the importance in big outbreaks than mosquitoes. But this is only a guess at this point of time based on the basically biobehavior of the mosquitoes and midgets. But you should avoid both. But since we could not so far do anything with the outbreak, is the fact that mosquito net was not very effective. And most of the screens that you put on your window, they are good for not letting mosquito to come in, but unfortunately, midget can easily pass through. They need very specific mesh to put on the windows to prevent them to come in. I think the midget means the small flies. In native language means small flies, very tiny flies, and they're really tiny. Any other question? My advice to you people, young medical students. I love medical students, obviously, you need really a start. Bring in these questions, crystallize these questions into null hypothesis type, and then come together as a group and really start tackling these simple problems, but meaningful, meaningful information can contribute to the literature. You can go next year to one of the big conferences in the United States and say, listen, I do know epidemiology of the Oropouche virus in Dublin. It's not really very hard. You need to have a good sample. You have people like Dr. Wong, the other people who can help you. I left one of you in matter of fact, young, basically faculty today. Uh, it's experience in clinical research, for instance. So, but ask questions. Ask questions. Because a student needs to ask questions. 100. Whatever sentence you hear, put a question mark in front of it. Whatsoever I told you today, in 3 weeks, 50% of them they are wrong. Believe me. Because the science of this virus is in infancy. If anything, I'm teaching you about something that you believe as truth. Wait for 50 years. 50% of it is not truth anymore. Science is in fluidity, is moving, and have very inquisitive mind. Ask questions. Thank you very much.